we're going to be able to, by the end of this podcast, list the phases of meiosis 1 and 2 and describe the events characteristic of each phase. And then importantly, make sure that you can talk about 3 events that occur in meiosis 1 but not during mitosis. We're going to compare meiosis versus mitosis. And a point that you need to understand is that no chromosome replication is going to take place at the end of meiosis 1 before meiosis 2, because the chromosomes have already been duplicated in the S phase of interphase way before this process even began. Here's meiosis 2. It occurs in 4 phases, very similar to mitosis. In fact, some would argue that it's almost a mirror image of mitosis. You've got prophase 2, metaphase 2, where the chromosomes are coming together. The nuclear envelope is disintegrating. The chromosomes are coming together on the metaphase plate. If you notice here, the mitotic spindles are grabbing onto the kinetochores, and they're going to split the sister chromatids, one in each direction. On anaphase 2, you can see that splitting occurring, and then in telophase 2 and cytokinesis, just pretty similar to mitosis. But the point is that 4 gametes are created, and then in each gamete there's just a haploid set of chromosomes. There's 4 daughter cells here. Another point to make is that each daughter cell is distinct from each other and the parent cell. There's been this mixing up of the DNA through crossing over and through independent assortment on the metaphase plate in meiosis 1, and therefore none of the DNA in any of the daughter cells are exactly the same. Which phase is represented in this cell? I'll ask you tomorrow. Take a look at that. And then diploid cells may undergo either mitosis or meiosis. Can haploid cells? Why or why not? Well, let's look at mitosis versus meiosis, and specifically mitosis versus meiosis 1. There's going to be some big differences here. I see this chiasma being formed in meiosis where crossing over is occurring. I also see lining up of these homologous pairs of chromosomes in meiosis 1 next to one another rather than on top of one another like you see over here in metaphase of mitosis. That's a difference. Additionally, they're lining up at different sides. You can see here that the red chromosome is lined up on the left here, but very easily this red chromosome in the middle could have been lined up on the right. It's just all due to chance, and that's going to lead to variability way down here in the daughter cells of meiosis. You're going to see this table that summarizes mitosis versus meiosis. Make sure you spend some time here. It just basically goes over what we just talked about, as well as these couple slides. 3 events are unique to meiosis, and they all occur in meiosis 1. What are they? You can press pause and quiz yourself right now. Synapsis and crossing over. You'll see number 2 is at the metaphase plate. There are paired homologous chromosomes instead of individual replicated chromosomes that are lining up in an independent assorted manner. And then in anaphase 1, the whole chromosome, this whole sister chromatid having homologous chromosome gets pulled over. It's not that the sister chromatids get split. That happens in meiosis 2, but in meiosis 1, the crossed over or sister chromatid, actually the whole X goes over, and you can see that if you look at the phases of meiosis 1. And you can kind of see that here, that the whole X or replicated chromosome is going to go over. Please perform question 261, sorry, question 10 on page 261. Here's the answer that you might have gotten. Maybe go back in the podcast and press pause so you don't see the answer. This will kind of test your knowledge so far on this process. Do you know what a feeder hair extension is? I don't either. I'm going to show you the objectives, make sure that you can answer these, and then I'll play a few movies, and we'll see if they work on the podcast. So go ahead and press pause here and answer these for tomorrow.
The cell prepares for meiosis just as it does for mitosis. But as the DNA condenses, each chromosome finds its homologous counterpart. These matching pairs of chromosomes hold equivalent genetic information, one set from mother and one from father. Next comes the first shuffle of nature's genetic deck. The two homologs trade genes in a process called crossing over. This forms new hybrid chromosomes. The spindles then place maternal and paternal chromosomes randomly on each side of midline. It is another shuffle that can lead to a host of different genetic outcomes. Finally, spindle fibers pull the homologs apart. This leaves the sister chromatids intact. With twice the amount of genetic information the cells will need, nature must cut the genetic deck in two. This second division produces 4 unique sets of chromosomes. Because they carry half the genetic information, we call these haploid cells. Many of them will mature into sperm and eggs, the reproductive cells called gametes.
In prophase 1, the DNA coils tightly and individual chromosomes become visible under the light microscope. Homologous chromosomes become closely associated in synapses, and they exchange segments by crossing over. By metaphase 1, the nuclear membrane has disappeared, and the microtubules form a spindle. Spindle fibers attach to only one side of each centromere, and the two homologous chromosomes attach to microtubules orienting from opposite poles. Each pair of homologs then lines up on the metaphase plate. Either maternal or paternal homolog may orient toward a given pole. In anaphase 1, the microtubules of the spindle fiber shorten and pull the chromosomes toward the poles, taking both sister chromatids with them. Each pole ends up with a complete haploid set of chromosomes consisting of one member of the homologous pair. During telophase 1, the nuclear membrane reforms around the daughter nuclei. Each daughter nucleus contains 2 sister chromatids for each chromosome attached to a common centromere. Because of crossing over, the 2 sister chromatids are not identical. During prophase 2, the nuclear envelope breaks down and a new spindle forms. In metaphase 2, spindle fibers bind to both sides of the centromeres. During anaphase 2, the spindle fibers contract and the sister chromatids move toward opposite poles. In telophase 2, nuclear envelopes reform around the sets of daughter chromosomes. Prior to meiosis or mitosis, DNA replication occurs. In both meiosis and mitosis, the chromosomes condense and become visible under the microscope. In meiosis, homologous chromosomes pair up or synapse, and crossing over takes place. Homologous chromosomes then align themselves along the metaphase plate, and each homolog is drawn to the opposite end of the cell. In meiosis, a second division occurs, resulting in production of 4 haploid cells. A single-cell division occurs in mitotic cells, resulting in 2 diploid cells.